All right, everyone. Good afternoon, good morning, good evening, depending on where you'll be joining us today. It is my pleasure to welcome you to another session of the Connecting Minds to Progress AI in Medical Image seminar series, Compass, where we discuss about medical image computation and advanced techniques on its analysis with the aim of improving healthcare outcomes. Today, we are joined by Dr. Ninon Burgos from Paris, specifically Paris Brain Institute, France. Dr. Ninon Burgos is a CNRS researcher at the Paris Brain Institute. She's the co-head of the Aramis Lab and a fellow of the PRIRI, which is the Paris Artificial Intelligence Research Institute. She completed a PhD in 2016 at University College London. In 2019, she received the ERCIM Paul Bayen Young Researcher Award. Our research focuses on the processing and analysis of medical images, on the use of images to guide diagnosis, and to the application of these methods to the clinic. In particular, she has contributed to 1, anomaly detection using both traditional image processing techniques and deep generated methods. 2, the translation of machine learning approaches for quality control and computer-aided diagnosis into clinical practice. 3, reproducible medical image processing and computer-aided diagnosis with machine learning. And 4, the development of open software. These efforts aim to enhance the reliability and scalability of research in clinical neuroscience and support practical advancements in medical imaging. Today, she will be teaching us and educating us more about AI and neuroimaging for the computer-aided diagnosis of neurodegenerative diseases. Ladies and gentlemen, please give welcome to Dr. Ninon Burgos as she starts her presentation. Thank you.
Well, thank you very much for the introduction and the possibility to present my work. It seems that I'm not allowed to share my screen.
Yes, so I'm fixing that right away. Now you should.
Yes, indeed. Let me find the good screen to share. And here you should see the slides.
Yes, I can see the slide now.
Perfect. Okay, well, let's start then. So yes, today is about AI and neuroimaging for the computer-aided diagnosis of neurodegenerative diseases. So what are we talking about? So medical imaging plays an important role in the detection, diagnosis, and treatment monitoring of brain disorders. So neuroimaging includes different modalities such as X-ray computed tomography, magnetic resonance imaging, so MRI, positron emission tomography, PET, and single photon emission computed tomography, or SPECT. In the following, we will mainly analyze the T1 weighted and FLAIR MRI, and also PET, particularly FDG and amyloid PET, as these modalities have been widely used in the context of neurodegenerative disorders, such as dementia, which is the focus of this presentation. So let's briefly talk about dementia. Dementia refers to a group of disorders that are caused by abnormal brain changes that trigger decline in cognitive abilities that are severe enough to impair daily life. So dementia affects behaviors, feelings, relationships. These are progressive diseases. This means that cognitively normal subjects can start with mild cognitive impairments that may or may not progress to dementia. Alzheimer's disease is the most common cause of dementia and will be the main focus of the presentation, but others exist. You have a non-exhaustive list here. The diagnosis of Alzheimer's disease mainly relies on a clinical examination and on cognitive tests, but imaging can play an important role. You can observe neuronal loss on structural MRI. we also call this loss of neuron atrophy, and atrophy is usually diffused, meaning that it is not restricted to a particular region. But the hippocampi are often affected, and atrophy is also visible through the expansion of the ventricles. Yeah, sorry, I lost the zoom, it disappears. So neuronal degenerescence can also be observed on metabolic PET. So here, FDG stands for fluorodeoxyglucose, so glucose meaning sugar. And so it is used to see the early stage of neurodegenerescence. PET is also useful to assess if certain proteins accumulate in the brain. Here is the example of the amyloid protein that is known to be part of the Alzheimer's disease process. It's not the only protein involved. You also have the tau protein that can be observed with tau PET. But you can also study other like imaging modalities such as diffusion MRI, but also study like fluids, like the presence of different proteins in the CSF. So as mentioned previously, neuroimaging offers a quite unmatched description of the brain structure and physiology, but the information it provides is not easy to extract and to interpret. So a popular way to extract meaningful information from brain images is to use computational methods based on machine learning to provide estimates of the category of pathology in a patient. A really large number of machine learning and deep learning approaches have been proposed to classify and predict Alzheimer's disease stages, but only a few are translated to the clinic. And this can be explained by different factors. First, the lack of reproducibility that comes from a lack of data sharing, of lack of fully automatic data manipulation, and a lack of code sharing, basically. It can also come from data leakage, meaning that the use, so that some people use test data in the training process. So, well, it's kind of a problem because this biases the results. And so it prevents assessing the potential like value of the methods being developed. There is also a lack of generalizability of the approaches that have usually been developed in a highly controlled setting, and so that have like problems like generalizing to other types of data. The evaluation is usually not pushed enough to really know like the limits of the approaches. There's also a lack of interpretability of these methods, meaning the ability to understand why your machine learning or deep learning model actually took a given decision. And this is like a problem for acceptance, basically, and to know how far like the approaches can be trusted. There are some approaches that exist to make this the classification-based methods, let's say, more interpretable, but they also come with like many limitations. So, well, I think it's good to keep like pushing these topics, but currently it's difficult to really rely on interpretability approaches. And the lack of clinical relevance is here like listed as the last point, but it's actually maybe like the most important one is that in like the field you see a lot of people like trying, for example, to differentiate cognitively normal subjects from patients with Alzheimer's disease. And this task is really interesting when you develop methods, but in terms of clinics, it has no use, basically. I mean, it's not how doctors will do things. So well, main limitation, lack of clinical relevance. Okay, so an alternative to computer-aided diagnosis systems that predict like the current or future diagnosis is to detect anomalies without an a priori of the disease you're aiming to diagnose. The image that contains then the anomalies can be used to predict the diagnosis if it's what you want to do, or just like better like characterize your image. And so in this presentation, we will focus a lot on this type of approaches. Here what I present is like images from MR images, but actually we are interested in the following, in detecting lesions in PET images. The approach consists of generating a subject-specific model of healthy appearance for the targeted imaging modality. And then we can compare this subject-specific model of healthy appearance with the real image of the subject. And by comparing the two, we can see the anomalies. And so this generates an anomaly map. I first developed a method using classic image processing techniques, like quite some time ago. It is based on a database comprising pairs of PET and MR images of healthy subjects. And it uses registration techniques, propagation, and image fusion to generate a healthy-looking PET based on the morphology of the subject under study. The healthy-looking model can then be compared with the real image of the patient to generate this anomaly map by computing a Z-score, for example. I'm going to show you a few examples of results. So here it's the case of FDG PET, so that shows you like brain metabolism and so early signs of neurodegenerescence. So we'll first start with a cognitively normal subject, so a healthy control. Here you have the FDG PET image of this subject and the corresponding anomaly map. And so here, basically, each voxel, the value of each voxel corresponds to a Z-score. And so here you see only very low Z-score, so no anomalies, which is normal for a healthy control. When you enter the early stages of mild cognitive impairment, you start seeing some small lesions here, so hypometabolism in this part of the brain. When you're in the late stage of mild cognitive impairment, you still see this hypometabolism in this region and also in this region. And this is really, it's where you expect hypometabolism in patients developing Alzheimer's disease. And finally, at the Alzheimer's disease stage, you still see like hypometabolism in the same regions. The nice thing is that the approach is not limited to like one type of PET images. It can also be used with other PET tracers. So here it's a type of tracer that shows you the accumulation of the amyloid protein in the brain. I'm showing you the same subject. So here you see that the PET image looks quite different from what we were seeing previously. Here still not much anomalies. And here you see the anomalies in red appearing as we go into like more severe stages of dementia. So the approach is not really PET tracer specific. It's also not disease specific, because you're trying to just detect anomalies, no matter the disease. And it's actually like the strength of these approaches is that you don't make any a priori on the type of anomaly that you want to detect. And so here I'm showing you three examples of a different type of dementia that is called primary progressive aphasia, that comes in different subtypes. So the agrammatic, the logopenic, and the semantic. And basically what you see is that you have anomalies. So here in dark blue, here is just a thresholded version of this image. You see anomalies in different parts of the brain, depending on the type of dementia, and this is normal. So really you detect lesions in different parts of the brains that are specific to like the disease you're studying. As you may have noticed, the method, if not the principle, is quite outdated. And so it's been replaced by deep learning approaches. Some closely follow the principle of generating a pseudo-healthy version of the modality of interest, so PETs in our case, from another modality. Here, T1 weighted MR images that shows the structure of the brain. So in this scenario, you can use, for example, a conditional generative adversarial network, so a conditional GAN, to generate PET images from T1 weighted images. And during the training phase, basically you only use images of healthy patients. So really you train your conditional GAN to reconstruct PET images of healthy appearance using only data from healthy subjects. Then in the application phase, pseudo-healthy reconstruction is obtained for the patient of interest and compared with the real image, again, as in the previous approach, to highlight the anomalies. Because here it works because basically the generator only knows how to reconstruct images of healthy appearance. So this is what it will do, at least in principle, no matter the input like image, it will output what should be the healthy version of a PET image that corresponds to this brain. And so when comparing this pseudo-healthy image with the real image, then you will highlight the anomalies. But then this is not really the type of approach we decided to focus on. Instead, we decided to work on a family of methods that only relies on the modality of interest for us, so it's still like PETs. A bit the same principle during the training phase, you only use images of healthy subjects. And so you train a network, so here an autoencoder to reconstruct pseudo-healthy images. So basically your network only learns how to reconstruct images of healthy appearance. Then in the application phase, you give as input the image of a patient with an unknown diagnosis. And because the network only knows how to reconstruct images of healthy appearance, then the network will output a pseudo-healthy image. But again, you can compare it with the input image to highlight the anomalies. One of the challenges that you encounter when you want to evaluate anomaly detection methods in the context of dementia is the absence of ground truth. So meaning that you don't have a mask of the anomalies as you could have when you study, like, for example, brain tumors on MRI. Here you just don't have a mask of the anomaly. So our first contribution was to develop an evaluation procedure that is based on the simulation of abnormal images to validate pseudo-healthy reconstruction methods. So here what you have, so same as what I've shown you like before. So during the training, you only use images of healthy subjects. And during the inference phase, then you give as input an image of either like an image of a patient with an unknown diagnosis, and you will reconstruct a pseudo-healthy reconstruction. When you want to evaluate the ability of, here, for example, we use a variational autoencoder, the ability of the VA to reconstruct pseudo-healthy reconstruction, what you can start with is check that when you give as input the image of a healthy subject, then you get as output an image that is very similar to the inputs. So you check that these two are pretty similar. So then in this work, what we did was to propose this simulation framework to where we can generate images with anomalies, but with known anomalies. So then what can we do? So we feed to the VAE this simulated abnormal image, and we get this pseudo-healthy reconstruction. Then what we can do is check first the quality of the reconstruction, because basically this image here that we reconstruct with the variational autoencoder should be really similar to the image of the healthy subjects that we use to generate the simulated abnormal image. So we can check that these two are the same. We can also check the extent to which the reconstructed image, so this one, appears healthy. And we can also check the ability to detect anomalies in here simulated images, but also in real data by comparing these two images. A bit more about the simulation framework. So basically, we start from images of healthy subjects. And it happens that we know the areas of the brain that are usually affected by disorders such as Alzheimer's disease. So we can design a mask for the areas that are affected. And then what we do to simulate the anomalies, we simulate hypometabolism by reducing the intensity within the mask. And so then it gives us, we can simulate different degrees of anomaly by more or less reducing the intensity within the mask. And so now the nice thing is that we have a pair of healthy appearing image and of an image simulating the anomalies. And so we have basically a ground truth now. And the nice thing is that we can simulate different types of dementia, because basically what's the difference between this type of dementia is where the hypometabolism is located. What we can do with that are different types of experiments. So here I'm first showing like an example. So basically we start with this image of a healthy subject. This is the mask that we use to simulate hypometabolism. So here this is the image with simulated hypometabolism that we get. And here is the anomaly map that we obtain with the anomaly detection approach. And so basically, this image, we should see like hypometabolism in this region that should match the mask that we use to simulate the anomalies. This framework allows us to perform different experiments. such as checking that when a high degree hypometabolism is simulated, severe anomalies are detected. So I'm going to explain you this graph on the right. Here, what you have on the y-axis is a healthiness. The healthiness is defined as follows. So it's basically the mean uptake that you have within the brain mask in the reconstructed, pseudo-healthy reconstructed image over the mean uptake, so the mean value that you have within the brain but outside of the mask. And so basically for a healthy image, so here that we call like ground truth, that should be around like 1. And then what you have here is the healthiness for the images where we simulate anomalies. And we simulate anomalies of only like 5% to 70%. So here we decrease like the intensity within the region, within the mask, about 70%. And so, of course, the healthiness index really decreases. And here, what you can see is that the healthiness increases in the reconstructed image versus the simulated image with anomalies. So what this means is that we, when we generate like the pseudo-healthy image, we are able to really reconstruct something that is pseudo-healthy, and so that has a healthiness like way higher than the image that presents anomalies. Ideally, we would like this to be at the level of the blue boxes, but what you can see is that when we give as input an image that has really severe anomalies, we have issues recovering like the initial image that should be here. But when we have not so severe anomalies, then we are way closer to our ground truth. And so this simulation-based evaluation framework really allows us to perform all these experiments that we couldn't do otherwise because we don't have ground truth. This framework in subsequent work, it allowed us to compare a large number of variational autoencoder variants, most of which had never been applied in the medical imaging context with like 3D data. So I'm not going to detail all like the variants of the variational autoencoder that we work with, but they are all listed here. And here again, we use the healthiness to evaluate and to compare the approaches. So ideally, you would want all this distribution of the healthiness to be close to this distribution of the ground truth. And so based on just this healthiness criterion, then the VAE variants that are the most like that perform like the best are basically the beta VA. Okay. Something that is really of interest to us is the fact of being able to handle intersubject variability. Because here you see that even among cognitively normal subjects, well, people don't all have the same brain. I mean, the shapes are different, the uptake, so the intensity can be different. And so, of course, when you're then interested in patients with Alzheimer's disease, then you don't want the variability in the brain shape to prevent you from detecting anomalies. And so we work on finding ways to handle this intersubject variability within the variational autoencoder framework. And so basically, what the PhD student here, Miley Solal, did was to actually study the variability that exists within the healthy controls that are used during the training phase. And so basically she can compute a standard deviation of all the PET images of the healthy subject that you have here. And then what we can do is, so here we have like the usual like anomaly detection approach where we give as input an image that may or may not have anomalies. It generates a pseudo-healthy image. And when we compare the input and the output, we get this residual map. And then what we can do is basically modulate this map using the standard deviation here. So, but yeah, that was computed using healthy controls. And so we end up with a map like this that is a bit similar to a Z-score, basically. So all this work was so the previous slides were using a lot variational autoencoders for pseudo-healthy image reconstruction. So they have many good properties, but they are also known to generate a bit blurry images. And so we've also been exploring other types of approaches. And so in particular, we've proposed an approach based on Bayesian flow networks that are basically based on diffusion models, but that introduce a Bayesian latent representation of the parameters of a data distribution rather than on the data itself. And so basically it really combines diffusion models and Bayesian process in how you progressively generate a pseudo-healthy image. I'm not going to go more into the details of the method because we don't have time, but I just want to show you an example of results. So here again, we used our simulation framework. So basically the ground truth here is the image of an healthy subject. From this image, we generate an abnormal scan using this mask of anomaly. And here, what you have are pseudo-healthy reconstructions using different types of generative models. So a beta VAE that was working well, but also a GAN approach, diffusion model approach, and what we propose. And here is the anomaly map that should look as close to this image as possible. And so we see that with the approach that we propose, we are really well able to get these areas, clear areas of hypermetabolism. Until now, all the work that I presented has relied on data from research studies. But we know that in real life, things can be very different. And so this is what we will explore in the following. So really our question is what happens to diagnosis prediction methods when we give this type of images as inputs, or to lesion detection methods when we give this type of images as inputs. And these images, I didn't pick particularly like awful ones, but this is what you have in like routine, so clinical care. And in particular, this is what you have within the hospitals of the Parisian areas. Many hospitals within the Parisian area, they are part of the same network that is called Assistance Publique Hôpitaux de Paris, so APHP for short. This network, it gathers 39 hospitals, so within the Paris area. And the very nice thing is that all the data that have been collected in these different hospitals, they are gathered in a clinical data warehouse. So it's in French, it's Entrepôt de données de santé, so EDS. This clinical data warehouse was created in 2017, and it's actually the first clinical data warehouse in France that was authorized to share data for research purposes. And so this warehouse gathers administrative data, medical reports, biological results, and imaging exams of more than 10 million patients. And so it means potentially several tens of thousands of brain images. Okay. So with this data, we actually worked on two projects. One was focused on diagnosis prediction and one on lesion detection. And so I'll start with the project on diagnosis prediction. For this project, we were working with T1 weighted MR images, and in particular 3D T1 weighted images. And so the first thing we had to do was actually to find these images of interest within the million of imaging exams that exists. And so this took quite a lot of work, just being able to identify the good sequences. So our inclusion criteria were quite simple. We wanted people aged over 18 and that had at least one cerebral 3D T1 weighted MR scan. And so this means a potential of 130,000 patients, but we had only clinical data for less than 20% of them. And so in the end, we worked with roughly like 10,000 images. The first thing we did was to perform quality control. We first started with manual quality control. So we basically annotated a bit more than 5,000 images. And what we observed was that already 1/4 of them could just be straight like reject, because either like they were not T1 weighted images of the brain, or because the quality that was so bad that you couldn't even distinguish a brain, or because the images were truncated, for example. For the images that passed this first like filtered, then what we did was to grade the quality of the contrast, the presence of motion artifacts, and the presence of noise within the images. So we had a 3-level scale. These image characteristics, they allowed us to define 3 global levels of image quality. So good quality, medium quality, low quality. We also annotated whether a contrast agent had been used when acquiring the images. So the contrast agent is called like gadolinium. So here is why you have gado, not gado. And basically what you can see is that, so as I said, 1/4 of the images are straight reject. Many are of low quality, and only a minority actually are very good quality. So this was whole like manual quality control, but then we used our annotation to then train neural networks to perform like automatic quality control. And so once we had work on the quality control, we could work on actually our task of interest, which was trying to identify patients with dementia. So we actually designed two tasks. So trying to identify patients with dementia from patients that had no dementia and no lesions visible on MRI. And this is a task that is really close to what you can find in a research context where people do Alzheimer's disease versus healthy controls. It's just that here we don't really have healthy controls because these are all people that had like an MR examination of the brain. So there must have been a reason why they had this exam. So they are not controls. The other task that we designed was to differentiate patients with dementia from patients that had no dementia but lesions visible in MR images. So typically people with tumors or stroke lesions. And we wanted to see whether the algorithms were able not only to identify pathological images, but also to recognize the type of pathology. Without further ado, some results. The classification performance, so the balance accuracy, was much lower within this clinical context than in a research context. So there was a 20-point drop in balance accuracy, basically. But maybe more interestingly, we observe that a difference in proportions of images with gadolinium, so contrast agents, and of medium good quality images can really bias the results. I will show you why in the next slide. And also, we used diagnostic codes. So these are the ICD-10 codes as labels to identify so dementia and the other diseases. And these are actually not robust labels. So the first point. So here you see like the cohorts that we used. So we had so 750 patients per class. So we had the same number of patients for all the classes. They have the same average age and the same sex distribution because basically these two classes were designed to match the dementia class. What is more interesting is the distribution of good medium quality versus bad quality images and the proportion of images with gadolinium. And here what you can observe is that the patients that had no dementia and no lesion, they tend to have images of poorer quality than the other patients. And what you can also observe is that people with dementia tend to have fewer images with gadolinium, which is normal because gadolinium is usually not used when you suspect dementia. But this, the neural network is using this information of the quality of the image, of the presence of gadolinium, to predict the diagnosis, and which is not at all what you want. And this is really a shortcut learning. It uses like a shortcut. So here's the quality of the images of the presence of gadolinium to predict the diagnosis, and it does not use really like the structure of the image, let's say, to predict the diagnosis. And so this is really not what you want. So we proposed a few solutions to avoid this problem. One is to generate images without gadolinium from images that had gadolinium, and this works pretty well. And about the quality, basically the only solution that you have is to only train such classifiers with images of medium and good quality, either from your routine clinical data or also from research datasets. But a challenge remains, and this is a lack of robustness of the labels that we use. They lack robustness because of several reasons. First, in France, they are used for billing purposes. And so this biases really the scores, and also they just contains like mistakes. And so these two things combined, they make like the labels like not very robust. And so that's why that in the following work, we turned to a task of lesion detection, which is unsupervised, and so we don't need labels anymore. For this work, we used another MRI sequence, this time the flare, still 3D, and we had to go through like the same process of finding actually this sequence within like all the data that were available. And so we work with a bit more than 10,000 images. We repeated the same process of first manual quality control. And here what we can notice is that we reject, like let's say from scratch, very few images. And the images are usually of good quality. And really you have almost like half of them of good quality, which was not really the case for the previous sequence, where the majority was more of bad quality. After the manual quality control, we also develop an automatic approach. And this allowed us to move to the actually task of interest that was detecting so age-related white matter hyperintensities in the flare images, so really a specific type of lesions. Same principle as before, we used only images of of healthy appearance, so without anomalies, to train a beta VAE, so that then we could feed an image with potential anomalies. The network will reconstruct something of healthy appearance. We could like compare the two to highlight the anomalies. Here are a few examples of results. So here you have 3x3 patients. They are stratified by FASECAS score. Basically, when you have a low FASECAS score, so of 1, you have few lesions. But when you have a high FASECAS score, so of 3, you have really more and large white matter lesions, what you can see here. And the results are quite promising. So in most of the cases, the approach is able to detect the anomalies. So here in purple is what is the anomalies that the approach like detected. Of course, we missed a few, and we detected a few that were actually not like lesions, but for a first work of anomaly detection on clinical routine data, it was quite promising. Here again, we are in the scenario where we don't have ground truth. We don't have manual segmentation of the anomalies. So what we did instead to validate our approach was use the FASECAS score as a proxy, because basically a high FASECAS scores means more lesions, so it means more lesional volume. And so we checked that when we had patients with a FASECAS score of 3, we would detect lesions with a higher volume than when we had patients with a FASECAS score of 1. And this is what you see here. We see indeed like differences. And the good thing is that this is robust to image quality. So no matter if the image is of good quality, moderate quality, or bad quality, we are still able to detect lesions that are meaningful. So in conclusion about this work, on the clinical data, so really quality control and dataset homogenization are crucial to enable translation of computer-aided diagnosis tools to clinical practice. Something to keep in mind is that not all AI approaches are appropriate for use in clinical practice. So, for example, the ones that predict a diagnosis, in our case, they were not appropriate because the labels we were using were just not robust, and because we had many biases, even though we tried to reduce them as best we could. And so even though we had a lot of images at the beginning, once you do the quality control and you homogenize things, then it's you don't have so many in the end. And so size at the beginning is not the only characteristic that matters about that. I will briefly conclude by mentioning that we, in the team I work in, we do a lot of open source software developments. So we have two tools, one that is called Clinica, that is useful to organize your brain images, to pre-process them, and one that is called Clinica DL, that is very useful to train a neural network when you are working with neuroimages, because it gives you like many tools to like manipulate the data sets basically. And so the two are pretty linked. Okay, so I will stop there, and just in conclusion, so basically by identifying brain anomalies at the scale of the individual, by developing the reproducible analysis pipelines that are in the software that we develop, and by validating our approaches in clinical data, we really aim to progress towards the application of systems for the computer-aided diagnosis of dementia to the clinic. But of course, there is still like a long way to go. So I would like to thank a few people. So on the left, you have all the PhD students that they developed the work that I presented, the software developers that work on the platform, and Olivier, with whom I've been working, and these are all my founders. And I thank you for your attention.
Wow. On behalf of everyone, we would like to say a very big merci beaucoup to you. You've really took us through the analysis and synopsis of about a decade-long work done between PET imaging and artificial intelligence, both the coding, both the image analysis. We sincerely appreciate you for taking the time to educate us on this. We have a few questions. I would like to start taking them one by one. One key question here, which actually came in first, was what's the difference between amyloid, tau, and FDG PET? I guess this person has been hearing those words loosely used around. I was wondering what could be the differences.
So if I here I have an example of the three. It's so basically the principle of PET imaging is that you inject a radioactive tracer to the patients, and so this radioactive tracer, it's built of two components, so an isotope that emits positrons and a molecule that is a molecule really of interest, let's say. So in the case of FDG PET, this molecule is something that looks really close to sugar. And so when you inject this tracer within the body, it will accumulate in areas that consume a lot of glucose, of sugar. And so in the brain, it will accumulate because the neurons, they consume a lot of glucose. And so it's interesting in neurodegenerative diseases because the neurons, before dying, basically they will start by consuming less sugar. And so this is what you can observe in FDG PET images of the brain. FDG PET is also used a lot to study like cancer, because tumors, they use a lot of sugar. And so you see them like that. For amyloid PET and tau PET, the isotope can be like the same, but the little molecule can change. And for amyloid PET, it will be a molecule that gets attached to the amyloid protein in the brain. So it gets attached to this protein. It means that it accumulates where the amyloid protein is, and so you can see the amyloid protein in the brain. And it's the same for the tau protein. It's just another protein that also accumulates in the brain. And basically, why we study that, we know that in dementia, the two proteins, amyloid and tau, they accumulate in ways that are abnormal compared with healthy controls. And so, and we know that depending on the quantity of proteins that you have and depending on where they are located, then it can give you like insight on like the progression, for example, of the patients towards a dementia or not. So that's why we can keep track of these different phenomena that occur like within the brain. So accumulation of tau and amyloid protein and decrease in glucose consumption. So that's why we can use these three tracers in the context of dementia.
Wow, thank you so much for that. I think that question is also similar to this question, but I'll still say it in case you want to share some further insight into it. This person is asking, what's the relationship between inflammation, metabolic activity, and neurodegeneration and tracers? I mean, it's similar to what you described earlier, but do you want to provide some?
Yeah. So because actually, so there's a fourth tracer that is a bit more like recent in the study of dementia that can actually show inflammation. Because the issue with like dementia and with Alzheimer's disease is that we still don't really understand fully all like the phenomena. I mean, we know that there is this accumulation of amyloid and tau protein that then leads to neurodegenerations. And we also observe like inflammation, but how all these things are related is still not like 100% clear. But there's really like no real interest in imaging like inflammation. So this is like the really current thing when you study like PET and dementia.
Thank you so much. We have shared that. Then there's another question here. What other neurodegenerative disease can AI-enabled pets be used to identify similar to dementia?
So if we take so for example, Parkinson's disease, in Parkinson's disease, you also have either like PETs, but also like SPECTs that is used. Actually here, I guess this is an example of Parkinson's, where you basically here see this lesion in like a control. You have this nice shape, like in a croissant-like, and in the patients you lose a bit. This part here is different, and so here you can also use like PET for that, and you can also use like anomaly detection approaches to detect that here you don't have like the PET uptake, the intensity that you should have if this person were like a control, but here it's a patient. So yeah.
I see. Thank you. And this person is asking, what are your experiences with clinical validations? Have you done any, maybe some clinical cohorts or deployments of these tools? Have you had any experiences with trying to bring them into the clinics? And what are the experiences you'd like to share?
Okay. Actually, I don't have experience like trying to have like a tool available really in the clinic. So there are several like reasons. Well, what you can achieve is have like a tool available in clinical research. So this can be not easily done, but done in the context of a research team, I would say. And so this, if you work with clinicians from, in my case, it would be like from the neuroradiology department, then yes, you can have like you can implement a tool that can be used by clinicians, but within a research context. If you really want a tool that is used in the clinic, then the process is very, very tough, and you need like to be like FDA approved or CE approved and everything. And so this goes really like, I think, beyond like what a research team can do. But then you can also like get in touch with someone from industry. Either you can create your own startup and like go for it, or you can just like have like a yeah, just like contact someone with industry, see if they are interested, and like co-develop something. But I think for a research team, it's too heavy.
So,
Yeah.
okay. Yeah. So I think we've almost on time now. We need to call it short, but before we go, can we kindly can you help us quickly answer this question? This participant is asking, can we use this imaging modality to predict future occurrence of dementia in younger patients too? And if the answer is a yes, what are the factors that would be part of our selection criteria for such kind of patient? Do you have any experience regarding this, like future occurrence of dementia?
Yes, actually, so this you can do in different settings. So, for example, when in the case where you want to basically, if I remove like all like the limitation from an image to predict a diagnosis. So this, I said that basically it was a bit like useless to just like predict Alzheimer's disease. But what can be more interesting is to see from an image if you can predict whether this patient that has mild cognitive impairment will or will not develop dementia. This is something you can do within the context of the framework of classification, let's say. But you can do things more interesting. You can, for example, try to predict the image of this patient in like 1 year, 2 years, 3 years. And depending on like the image that will be generated, whether it has atrophy or not, then you can infer whether they will like develop the disease. So this is based on images, but of course, there are other like types of data that are very useful for that. And so actually in my team, so this is not something that I do, but what my colleagues do, they develop models to, well, to actually model like the progression of the disease based either on like clinical scores, but also on features extracted from images. And so from this, you can then predict like the trajectory, the evolution of a particular patient. And so this can be useful at different levels, either like when you are in the setup of clinical trials, when you want like patients still at an early stage, but that should develop quickly like the disease so that your clinical trial doesn't last for like too long. So this can be useful. And it can also be useful to identify patients that could benefit from treatments, because the one that will like stay like basically stable and do not progress to dementia, then okay, just leave them like alone. But if you predict that some people will evolve quickly, then maybe these are the ones that you want to treat in priority. So there's a whole like literature on the topic, but
Yeah,
yes.
to figure it out. I completely
Yes.
agree. Thank you so much once again, Dr. Burgos. We sincerely appreciate your time, and we hope when next we call on you to come and give us another talk, you would give us a yes. Thank you, everyone. Before we call it a day, I would quickly like to update you that we'll still be having this seminar series next month rather, and the speaker is going to be Rohit Jenna from the University of Pennsylvania, where we'll be talking on the understanding the computational models for dense correspondence matching. And this is going to happen exactly the same third Friday of August, 12 p.m. EST, 16 via 100 GMT, as might be applicable to your time zone. Thank you, everybody. Do have a lovely weekend ahead and do enjoy the rest of your day. Bye.
Thank you all.